Hi everyone, today we'll be talking about the apoptosis signal transduction pathway, and it will be presented by myself, Loniki, Yeben, Shui Jasmine, and Puente Anissa.
Apoptosis is a programmed death cell mechanism characterized by cytoskeleton depolymerization, cell shrinkage, chromatin condensation, and nuclear fragmentation. Apoptosis occurs during development and aging and is a homeostatic mechanism that maintains cell populations in tissues. It also is a defense mechanism in immune reactions or when cells are damaged by disease or noxious agents.
The mechanism of apoptosis has three different pathways leading to cell death. The first one being the intrinsic pathway, which occurs when the cell is under stress or has unrepairable damage. In this, BCL sensors detect this damage and signal to BAK and BAX proteins to form pores in the mitochondrial external membrane. This process is called mitochondrial outer membrane proliferation. Then, cytochrome C is released into the cell's cytoplasm and binds to APAF, which leads to apoptosis formation and activation of caspase 3. In the extrinsic pathway, ligand receptor interaction is required between the ligand FASL and its receptor located in the cell membrane. This results in the catalytical activation of procaspase 8 and procaspase 1, which then activates caspase 3 and caspase 7. Lastly, the perforin pathway involves the cytotoxic T cell recognizing its target cell through receptors, which induces the secretory lysosome exocytosis. Perforin then opens pores on the cell membrane to allow entry for granzyme A and B. They then activate procaspase 3 and initiate the caspase cascade to reach caspase 3. In this diagram, we can see how both the intrinsic and extrinsic pathway lead to caspase 3 activation. The reason why caspase 3 is so important in this pathway is because it's the main executioner caspase protein. It initiates the execution pathway for the final phase of apoptosis, ultimately leading to the formation of apoptotic bodies by blebbing.
Pathway malfunctions. So let's start with p53. p53 is a nuclear transcription factor that acts like the security guard of the cell. It constantly monitors the cell DNA for any damage. But when p53 is mutated, this guard can no longer detect DNA damage and doesn't just step aside. It actively inhibits normal p53 from working properly. Without proper detection of DNA damage, cells accumulate more mutation, and this unchecked damage can lead to cancer. Next, we have the BCL2 protein family. These proteins are key regulators of apoptosis, and when cells are healthy, BCL2 protein prevents apoptosis from happening unnecessarily. However, when overexpressed, BCL2 protein can become problematic. In damaged cells that should undergo apoptosis, BCL2 overexpression prevents apoptosis, allowing these harmful cells to survive and multiply, leading to cancer. Last, we have caspases. Caspases are enzymes that drive apoptosis. There are two types of caspases, initiator caspase and executioner caspase. Initiator caspase, when it's inhibited, cells cannot go under apoptosis. This leaves damaged and mutated cell in the body. On the other side, if the executioner caspase are overactivated, they can kill healthy cells. So in summary, mutations in p53, overexpression of BCL2 protein family, and malfunctioning caspase activation all play a major role in cancer development by either allowing damaged cells to survive in the body or healthy cells to die.
BCL2 inhibitors are targeted therapies designed to block the anti-apoptotic BCL2 protein, allowing cancer cells to undergo apoptosis. In the diagram, image A shows BCL2 binding to BIM to promote cell survival. Image B shows venetoclax, a BCL2 inhibitor, disrupting this process and triggering apoptosis by activating pro-apoptotic proteins like BAX and BAK. Venetoclax has shown strong clinical results, especially in chronic lymphocytic leukemia, by promoting cancer cells' apoptosis, making it an innovative intervention. p53 gene therapy restores the function of the p53 tumor suppressor, regulating cell death and DNA repair to prevent cancer growth. It triggers apoptosis through both intrinsic and extrinsic pathways, activating caspases to induce apoptosis in cancer cells. Gendesine, the first approved gene therapy, delivers a functional copy of the p53 gene, helping cancer cells regain the ability to restore and undergo normal apoptosis processes. PD-1 inhibitors such as pembrolizumab restore T-cell activity and prevent exhaustion, allowing the immune system to recognize and attack cancer cells. These inhibitors enhance immunity by reactivating immune cells and promoting apoptosis in cancer cells. Pembrolizumab induces programmed cell death in cancers like melanoma, helping the immune system eliminate tumor cells and providing significant benefits.